Hello everyone, I hope you're doing well. My name is Céline Barbib. I'm a PhD student at Institut Pasteur de Tunis, and I will be your host this evening. Thank you so much for joining us for our first webinar organized by Young Researchers Association of Institut Pasteur de Tunis and Institut Pasteur de Tunis. Actually, we try to support, promote, and disseminate the scientific culture to bring citizens and students closer to research. I'm so happy to present one of our brilliant Tunisian young researchers, Dr. Houssem Eddine Othman. Thank you again for accepting our invitation. I will briefly introduce you to our audience. So you're a bioinformatician from Sydney Brenner Institute for Molecular Bioscience, which is part of East University in South Africa. You earned your PhD degree from the University of Carthage under the supervision of Professor Najed Sraili at Institut Pasteur de Tunis. Your graduate work includes studying the structure-function relationship of anti-cancer peptides from scorpion venom, and currently you're doing your research in pharmacogenomic characterization from high-throughput sequencing data in sub-Saharan populations. You're also developing new bioinformatic and machine learning tools for genome annotation applied in science and precision medicine. So before we start, I invite all the participants to leave their question for the last part of this webinar, and you'll also find an evaluation form, the link in the comment section. So please feel free to share your opinion about this initiative. It will help us improve our next event. So, Dr. Houssem, we know that science during crisis helps guide decision-making, and it's one of the active fields to promote knowledge during any outbreak. But the COVID-19 pandemic has resulted in a mass of academic papers, either published or post-printed. We can even talk about speed science. And the data surrounding the biology, epidemiology, and clinical characteristics are growing daily, which is normal, but it can generate misinformation. And the fact that you can share this information with a simple click can generate fears. So today's topics is science vulgarization, social media, and bad research in the time of COVID-19 crisis. So please, Dr. Houssem, let's hear from you.
Thank you, Céline. Thank you all for having me this evening. I hope that everyone is holding well in this lockdown. It's been exactly 1 month since I haven't walked outside my apartment, and so I have a lot of time to think about different matters, including the coronavirus crisis and how it affected our lives, first as citizens, but particularly as scientists. So I had a very nice introduction from Céline. You know all that I did my graduate studies with Professor Najed Sraili at Pasteur Institute of Tunis, and I'm currently working at the Sydney Brenner Institute for Molecular Bioscience. We have a very nice historical building and a beautiful garden where we can take our group photo and do our social events. We are researchers from different countries working on genetic studies, including high-throughput sequencing analysis, GWAS, bioinformatics development, with applications in anthropology and disease determinism studies. I'm personally a member of GSK South Africa Collaboration Consortium, led by Professor Scott Hazelhurst. Our main objective is to characterize the genetic diversity of genes that are associated with drug metabolism. That said, I'd like first to say that even though it appears that this talk is about coronavirus, its main idea can be projected on other topics, as we are expecting a significant impact on our way of thinking about the role of science in society. And in fact, COVID-19 outbreak would not be the last health crisis that the world would experience in the upcoming years. We're expecting actually new viruses to emerge due to the demographic changings in the world. And on the other hand, we still have unsolved issues like cancer, malaria, tuberculosis, that makes a great challenge for the humanity. And so it's very important to emphasize on the role of scientific research as the main driver for any rational decision making. And in the last couple of years, the science has been heavily challenged. So we have the anti-vaccination movement and global warming denial campaigns. These are a few of these challenges that show how the center of gravity about scientific issues has shifted toward politicians, lobbying partners, and advocacy groups. And these are actually matters of human existence. So the new COVID-19 outbreak made up a great opportunity for scientists to reclaim their role in the society. And we are currently around 2.9 million cases worldwide. We are experiencing widespread of coronavirus in low and middle income countries, including Africa, Latin America, and some other Asian countries. And so the situation, the crisis situation, incite people to go after the scientists so that they can give solutions for them. And the outbreak actually is a tangible example of how scientific research can provide answers to a life-threatening situation that should be tackled in immediate time. And it's actually very difficult to explain to someone why you are doing an experiment on mitochondria or specific for a pathway, or why you're doing an infrared spectrum measurements for a molecule. So it's very hard to explain that to people. It would not help you either to say that I'm doing science because of the knowledge, because science is illuminative at first. But what can help in the crisis of COVID-19 is that you can say that whatever it is with this coronavirus, we can study it, understand it, get it, and save your life. And so a lot of efforts have been made to tackle the problem of coronavirus from all the phases. And the role of researchers in this context can be summarized in providing insight into the origin of the virus, making genome analysis, providing fundamental knowledge to explain clinical outcomes, repurposing and reinventing new drugs to cure diseases, inventing new vaccines, and doing the mathematical modeling to understand the dynamics of the outbreak. And however, not so obvious, scientists actually are moral boosters, and this is maybe one of the outcomes of the specific outcomes of COVID-19 crisis. So you would probably trust someone that tells you that eating apples will cure cancer because a new scientific study has proven that. But we will get to that later in the talk. So at this point, it's worthy to remind you about the evolution of COVID-19 outbreak. I'm not going to give too much details here, but looking at the timeline, you may notice the increasing rate of scientific breakthrough about the subject. Just between the first case of COVID-19 in Wuhan, China, and the submission of the full genome of the virus, there is roughly less than 1 month. And it's obvious that science of this level needs the deployment of all tools to share data, publish results, and keep up with increasing demand on knowledge. So in other words, the peer reviewing system or process cannot handle this situation efficiently. And we have witnessed an extensive use of data sharing repository like GSAIS, for example, which is handling the genomic sequence data of diverse geographic origins. Some other repositories like the one managed by the Johns Hopkins University with their famous dashboard are harboring details about data about infections, death tolls, and other metadata. We also noticed an extensive use of social media platforms like Twitter, Slack, Facebook to allow efficient communication within collaborative networks. But perhaps the most iconic tool for scientific research at the time of COVID-19 outbreak are the preprints. And arguably the most known of them are bioarxiv, medarxiv, and chemarxiv. So the process of paper submission to a preprint is very different from peer-reviewed journals. Basically, after you prepare your manuscript in a typical structure, you send your paper to a preprint platform, and within a couple of days, the manuscript will be available on their website. So there is no peer reviewing process. And preprints allow you to circulate your review results within a large scientific community while avoiding the lengthy process of peer reviewing, which I think it is critical for knowledge dissemination about SARS-CoV-2 and COVID-19. And in addition, preprint platforms offer the possibility for scientists to evaluate the transparency for submitted results. They can give you feedback, they can point out to weaknesses in your study, and they can suggest improvements. And the last thing is that the preprints are the property of preprint is the property of the author, and he can choose the type of license for sharing its content, which cannot be restricted by publishing policy, even after the submission to a peer-reviewed journal. And as you can see, preprints were the choice of publishing for papers about COVID-19 within the 2 months, the first 2 months of the outbreak, surpassing even the publication categories. So you can also see from this figure how social media are implicated to spread the new scientific findings. Typically, you wouldn't have more than thousands of tweets per paper. Some of the papers are about fundamental questions like the origin of the virus from, and they profit from a wider coverage rather than some others. And in all of these, all of them, there is one preprint that has been tweeted more than 17,000 times, and it will be the subject of the next discussion. So the authors of the preprint claim that there are insertions from HIV-1 virus in spike protein of SARS-CoV-2 that haven't been observed in SARS-CoV, the previous virus responsible for the outbreak of 2003. So, and the only version of the preprint now available is in archived version, is an archived version, because the authors withdraw it from bioarchive. I would like just to make some disclaimers here, because I'm not refuting the hypothesis of conspiracy theory. And actually, it is maybe safer to deny the conspiracy theory. No one wants to be called crazy, but even this level is just a derogatory term for what we call in science human manipulation of the virus genome. And biological warfare existed at the time when the Mongols attacked the walls of Afa in Ukraine, and the conquistadors in New Spain, nowadays Mexico, used it to spread smallpox among Native Americans. And last but not least, we have witnessed also the crisis of anthrax attacks in 2001. So it is still a theory, and no prejudgment should be made before backing the claims with scientific evidence. And this goes both ways. So the work on this preprint consists of aligning the spike proteins from SARS-CoV-2 with SARS-CoV first, from which they noticed 4 insertions in the former. They looked to the sequence of these insertions, and then they blast them, and then they found that they resemble substrings to in GP120 and GEC proteins from HIV-1. Then they built the 3D structure of the spike protein to show the location of these insertions. From that, they have concluded that this is a deed of a human manipulation for some unknown reasons. There is, however, a major drawback in their analysis. And if you actually took the sequences, and I did that, I took the sequence of these inserts and did an homology search using BLAST against all the viridae, and you'll find, if you try that yourself, you'll find that the levels of uncertainty to define a hit is very high, and this is quite expected for short queries. And if the e-value score, for example, is very high, the probability that the hit is obtained by pure chance is almost certain. And you can see this from this graph, because naturally for the situation of these inserts, we are at this level, being that the uncertainty is almost the obtaining these results by pure chance is almost certain. And moreover, when I did the blast, other sequences emerged at the top of the report, like the remnant lentivirus, hepatitis B virus, and some other phages. So it's not really clear why they stick themselves only to sequences from HIV-1. And more than that, actually, the authors did not report to which of the HIV entries the insertions are belonging. Either also it is a property, the property is not general, or if it's not really clear that this property is general, or if it is unique for some isolates of HIV. And I looked for them in both the GP120 sequence submitted to GBank in this sequence, and another sequence of GP160, which is the precursor of GP120, and I haven't found any trace of these insertions. Finally, the authors say that these insertions are part of the interaction site with the host cell. They haven't mentioned the target protein, but I'm going to presume that it's ACE2. And if that's the case, the insertions are way far from the protein-protein interface, as you can see here, because we expecting the insertions to be at this level, but as you can see, this is the interface, and none of these insertions are localized there exactly. So recently we have submitted a preprint, which is currently under peer reviewing, about the interaction of a spike protein with ACE2 expressed on the surface of host cells. And this is a collaboration between scientists from Sidney Brenner Institute and Pasteur Institute of Tunis. And I like first to thank all of them for their contribution. So in this work, we focused our analysis on the protein-protein interface between the RBD domain from the spike protein and the ACE2 human protein localized on the host cells. And in our results, actually, we noticed that there is what we call the swapping, the clade swapping phenomenon, meaning that the sequence from the current COVID-19 outbreak, they clustered with differently if you use the full genome of the isolates, and if you use also only the RBD sequence, which is implicated in the interaction with the host cells. And what we have seen actually is that there is a large energy jump, meaning that if we measure the interaction energies, we found that there is a big gap between what is supposed to be the progenitors forms of the current virus, which are the C45 and the C21 isolates, and with the current virus, which is this one here. And as you can see, the energies are quite different between these two groups, and that actually points out to that there was a major drastic events that will allow this jumping in the energy. So there is a very big gap that we don't observe currently in the available isolates that allow to see this phenomenon. And on the other hand, our analysis on the protein interface showed that the SARS-CoV-2 current form is not really very different from other forms, because if we expected really that this is a human manipulation in some level, then we expected this node to be very different from the other nodes, but we haven't seen that actually. And let's suppose that if I am an evil scientist and I want to destroy the humanity, even with that, I think the most obvious choice here, and just keep this as a secret, if I want to render the virus more lethal, or at least increase the interaction of the virus with the host cell, I would probably change the amino acids here, E484, with probably a polar amino acid, so that I can avoid the incompatibility between the two charges. And actually, this interaction, you see it right here in the histogram, which is very unfavorable for the virus. So what would be the consequences of this study? Actually, I mean the Indian study. So the most obvious one is that exploiting these findings justify racist behaviors, which would increase social instability, and it doesn't have to be explicit in nature. People actually ceased to go to Chinese restaurants because of the COVID-19, and in some extreme cases, violent attacks have been recorded in different countries. The other outcome can actually affect the social morale. Remember that scientists are the antidote for fear and the shield against social anxiety. And the Braden study has not been popular in the scientific community, not because of its conspirational nature, but because of the flaws in the methods and the interpretation of the results. And that has been the subject of an extensive and transparent discussion in bioarXiv platform, which has led the author to withdraw the paper. So when Professor Luc Montagny brings up the same subjects again in TV plateau, that would increase not only the social anxiety, but the mistrust in science. What also things up is that Montagny's claim coincide with Donald Trump's accusations for China and the World Health Organization as a distraction for his failed plan to fight the outbreak in the US. Now for the second example, the issue starts from a claim made by an aviator in a blog that hypoxia and not acute respiratory distress syndrome or pneumonia is the main cause of death in COVID-19. And from there, everything went wrong, really. And even though very few clinical observations backed up this claim, it went very viral on the internet and the social media. And the situation has worsened after the publication of this preprint in chemarXiv, who proved, and I put proved here under quotes, that SARS-CoV-2 blocks the access of oxygen in hemoglobin. And for that, really, I apologize because I cannot maintain my scientific neutrality for this paper. All the elements of in the paper are very bad, and it's a very typical example of a bad research. So before diving into complicated things, I ask myself honestly, and it's an innocent question that any undergraduate student would ask, why would the virus risk its chances of survival to infect a type of cell that with no replication machinery? And so that's the first question. And in summary, for this paper, the authors of this study did a computational analysis. It's a purely in silico study. They took 6 viral proteins and they did the docking with porphyrin, which is the like the heme group, to prove that the inhibition of the hemoglobin is done by the SARS-CoV-2. So the first error, you don't make the docking to prove that a binary complex is formed between a ligand and a protein. You can even try this by yourself. Take any molecule with no affinity toward the receptor, do the docking, and you would get a complex which is not realistic at the end. And you will even get a measure of the estimation of the binding energy. And so at best, the docking would be useful to filter some hundreds of lead compounds from thousands or millions of ligands, an experience that we call virtual screening. And really, in its ultimate goal, the docking would tell you what is likely the likely complex if a ligand and the receptors are known to physically interact with each other. So you have to know that information a priori. Second, the authors used a protein-protein docking tool to solve the problem of a small ligand interaction. And in protein-protein interactions, Partners are relatively rigid. They interact mainly by shape complementarity. They engage Jar-flat interfaces, and really you have really big complexes in there. And however, in ligand-protein interaction, ligands are highly flexible, and they both the electrostatic, the complementarity effect, and the dissolution effect, and the quantum effect are important for the interactions. Besides, the ligands tend to interact with cavities within the protein, and they are highly flexible. All the imparted elements in ligand-protein dockings are poorly modeled in protein-protein docking. So they are really using the wrong tool to solve their question or to answer their questions. And last, they have presented astronomical values for the prediction of the interaction energy. And to put things into perspective, if you take a protein, a fully folded protein, and a fully extended protein, a fully unfolded protein, the difference in the energies would be at maximum 10 kilocalories per mole. And as you can see, they are reporting here values of billions and millions, the only explanation for that is that they have severe atomic clashes that cause the van der Waals energy to be very high. And as you can see, the van der Waals energy at this level here, it indicates that there is a severe clash. And if you have a severe clash, then you cannot see any effect of any biologically relevant interaction. The last example, I wouldn't say it's a bad science, but more or less a failing to comply to scientific norms, and how also mediatization can worsen things up. We all remember Prof. Didier Raoult and his famous interview in which he defended the usage of chloroquine or hydroxychloroquine. So after that, the pressure has increased on the French government to approve its usage, despite the fact that there are no conclusive studies available so far about the chloroquine. And the massive trending about the drug after Trump's endorsement is believed to cause death and poisoning in some individuals. And one potential problem, however, that has not been or has been overlooked by the clinical studies, including that of Didier Raoult, is the G6PD deficiency, which can cause acute toxicity when you use chloroquine or hydroxychloroquine. It might not be very relevant in some regions in the world, but in sub-Saharan Africa, for example, G6PD deficiency is frequent. And G6PD actually is an enzyme that eliminate the reactive oxygen species that can be caused by the reaction of chloroquine with the heme group of hemoglobin. So this is our new study that should be available in the next couple of days, in which we showed that the distributions of variants associated or that cause the G6PD deficiency is significantly different even between neighbor countries. Even like, for example, between Burkina Faso and Benin, or Botswana and South Africa. We also showed that there might be other deleterious variants that would cause the deficiency, and hence any treatment protocol should consider the genetic diversity before making any plan to use chloroquine or hydroxychloroquine in sub-Saharan Africa. So finally, I just wish to emphasize the following points, which I think would help to build trustworthy scientific speech with society. First, it matters to choose our word in communicating science. Being subjective and judgmental or excited about something would not be helpful when things go wrong, and things really actually can go wrong, especially that we are living at the transition between two eras in biology. The one we are currently witnessing is characterized by an increase in complexity of scientific data. And whenever something becomes complex, errors can pop out everywhere. So this is the main issue. And in the new era also of biology, this new era is characterized by increased importance of reproducibility over the originality of the discoveries. And in other words, it doesn't matter if you found a new insertion from HIV-1 in the coronavirus genome, as long as nobody saw that but you. So the second thing, I think that as scientists, we are in the business of the unknown. We like it when we don't know things, because that drives our motivations toward solving questions. I cannot find any solution on how to make the vulgarization of this property, and this probably should be discussed in the future. What we should know, however, is that it defines our personality among other groups like physicians, managers, and engineers. And I think we should not forget this, even when we think to claim our lost stature in the society. And third and last, my personal opinion is that science has built an efficient framework to communicate its benefits for the society. We call them articles, communications, posters, academic eras, academic institutions, and others. But the popular buzz culture is not one of them. But we have also to admit that mediatization can help to increase the awareness about science. And for that, I also have no answer on how to keep the balance between scientific objectiveness and advertising science. And with this, I end up my talk. Thank you for your patience, and stay safe. Thank you.
Thank you so much, Dr. Hussein, for this presentation. So now the questions, we have a lot of questions, that's cool. The first one is how scientists should explain scientific facts or results in order to be more interesting than this theory. I mean, what's the actual role of a scientist in such uncertainty?
You mean for the COVID, especially for the COVID-19, or?
Yeah, or in general, but I mean, generally speaking, how can we explain and make the results and the facts more valuable than the other claimers?
Yeah, I have a really very personal view, but it's not necessarily many cannot share my view actually, because scientists are not positions at the end of the process. They are working on their lab, they in the shadows, and the main function of scientists, I think, it's to provide knowledge rather than thinking about application or any other thing. So I think we should, like I said, we should be satisfied about our role in the society, or this function is to provide knowledge to the society and the humanity before thinking of any other thing. The problem, probably the problem also in low and middle income countries, like in Tunisia or even in South Africa, that we are not also at the summum of the breakthrough discoveries. I think also the research and especially the COVID-19 crisis has proven that you can just copy and paste from France or from the United States, and the research, even the fundamental research, should be applied and should be reoriented toward the real issues of the local society.
Okay, so we have another question. What's your opinion about the 300 patient study and chloroquine concern?
Sorry, which study? Which study of that? Is it the
300 patient study and chloroquine concern?
Okay, I don't know really exactly the number 300. I know there are two. There is a Chinese study about the chloroquine, and there is also the Giroud study, and there is the Brazilian study about the chloroquine. None of them has provided really a firm answer of the efficiency of the chloroquine. And the problem is that, like I said, the doses they're using are way too high than the normal doses used for to treat the malaria. For example, I think in the Brazilian study they recommend the usage of 5 milligram per day twice. So it's 1,000 milligram per day for one patient. And the normal or the doses used for the malaria, I think there are 300 milligram per day. And actually the Brazilian studies has ceased and because of the toxicity. And I think also the Brazilian studies ceased because there is a very important component, African component within the population, and that probably is also can be due to the G6PD deficiency. So so yeah, if you can point me out to which study is that, and I can discuss that with you.
I think it's the placebo one, but I will check it out. Okay, so the next question is, which technique tool would you suggest to use instead of the choking tool?
Actually, I wouldn't suggest any tool, any computational tool in this at this level. Actually, this is a very critical point on defining the physiopathology of the virus, and also this is an issue of the modern biology, because it's really tempting to use computational tools to answer questions, but this is not the typical case in that. However, if I know that the hemogroup can interact with the virus, I would first do the experimental studies, the in vitro studies, binding studies, for example. When I am sure that the complex will occur, then with that I can do the docking. And for the docking, I can use there are many tools, free tools like AutoDock, Plants, Dock, all these tools that can be used for ligand protein docking.
Okay. So we have another question. It seems that several other viruses have similarity with HIV. Is it? So I mean, I think.
I'm not aware.
Same sequencing with the HIV.
Yeah, I'm not aware really of that, because all the studies that I've been aware of, they haven't mentioned the link with HIV one. But if there is a link, also it doesn't matter if you find the link or not. You have to explain it. You have also to explain it. And even like I said, when you say a link, it's not an evolutionary link. It should be some recombination event or an insertion or even a human manipulation. And that the human manipulation, I think they tend to involve fragments of big length rather than punctual mutations. So, but in the case of the Indian study, the insertions are not. We still haven't explained why there is these insertions and their role in the physiopathology of the virus. But these also insertions, we don't say firmly that they come from HIV, because they can also came from other viruses, even other organisms in mammals, because we found the same sequences in mammals and other inferior groups. And on the other hand, also there is the problem of the representativity of HIV sequences in the NCBI database. When you have a big representativity, then these sequences tend to pop out when you do your blast. So you will probably observe you will suspect a relationship between the two viruses, although it's not due to the a molecular event or a natural molecular event.
Okay. How do you see the relationship between science and policy after this pandemic?
Sorry, science and?
How do you see the relationship between science and policy after this pandemic?
Policy.
Yeah.
I think for now the science so far the science is science has driven the decisions or the major decisions about coronavirus, and you can see the reports of World Health Organizations, the daily reports. Actually, there are lots of studies and ongoing studies and updated studies. I think for now we have to, like I said, reclaim the position of science in policy in deciding the policy of the health policy of the country. But on the other hand, also I think in Tunisia, at least, the COVID-19 will change the health system in Tunisia because more fundamental studies will be involved. High throughput sequences, probably they will take their position in the diagnostic system. I hope that
and
also
the importance of research also. The importance of research. The importance of research in Tunisia also.
Yeah, of course, because I think that because so far there is, this is my own opinion, there is some kind of separation between the health system and the scientific research, and I think that now they started to approach each others. And I hope in the future, thanks to the new technologies, the high throughput technologies, there will be this tightening in the relationship. But also we hope that probably in the future to see biobanks and projects for biobanks, either for controlling the outbreaks or profiling the diseases in the country. Because like I said, we cannot, and this crisis showed it clearly that we can't copy and paste from other countries on Tunisian population or South African population or any other in the whole world.
Yeah, actually, you answered two questions at the same time, so that's cool. Okay, so science specialized reporters and journalists are the missing part of the puzzle. Don't you agree?
Like I said, I don't have the problem is not to find, because there are, even in Tunisia, there are scientific journalists. There are scientific journalists. The problem is that, like I said, how we communicate the science and the complexity of science to the public, and what we want exactly, and we have to define what we want exactly on doing that step, on what we want from advertising the science. Do you want financial support? Do you want the contribution from the society, not only from the government? And that will define how we are going to approach this question in the future. But also I agree that scientific, not only scientific journals are lacking in the country. I think also the dialogue in the common language of the country about science is lacking. We don't have so far, like in Derja, for example, we don't have a scientific dictionary in Derja, for example. And that would be actually a very interesting project.
Yeah, I think we have to prepare like new tools to present and communicate science to society. Yeah, for better understanding. Okay, I think there's no more questions though. Do you have any comments or any remarks or?
So far, no, I don't have comments. Hope everyone that it's safe all over the world, even Tunisia or all over the world, and keep doing science and enjoy. I think enjoy also these few days of free holidays.
Thank you so much, Dr. Hussein. Thank you for all our participants, and thank you for your questions. Stay safe and stay curious.
Thank you.
See you next time. Bye-bye.
Thank you, Cyril. All right. Bye.